is Cristal. I am PhD student in graduate program of Genetics and Molecular Biology and member of Bioinformatics Core of Hospital de Clínicas de Porto Alegre. Now I present my work entitled Cancer Pathways are Deranged and Mucopolysaccharidosis. Mucopolysaccharidosis of MPS is a group of genetic lysosomal storage disease when defects occur in the activity of lysosomal hydrolase involved in biosynthesis or degradation of glycosaminoglycans. Many glycosaminoglycans are so altered in a variety of cancers. Besides, in MPS, secondary cell disturbance affects pathways common to cancer. For this, the aims of this work are to identify ontology terms related to cancer in datasets of MPS available in public databases and to compare the ontologies in the different types of MPS analyzed. For this, we search datasets in GEO and in ArrayExpress with the terms mucopolysaccharidosis and the respective disease name, for example, for MPS type 1, Hurley disease. Next, we perform gene ontology analysis with QuickGo and KEGG annotations in TinyGo and Cytoscape with the ClueGo plugin. The statistical analysis was based on hypergeometric distribution followed by false discovery rate correction. The datasets obtained was 1 of MPS type 1, 2 of MPS type 1, 1 of MPS type 2, 1 of MPS type 3A, 3 of MPS type 3, 1 of MPS type 6, and 4 of MPS type 7. The organisms of these datasets are 1 dataset of dog, 8 of mouse, and 3 of human. The gene expression analysis was performed with appropriate packages of microarray and RNA-seq methods in the R environment, and these results and these plots are available in MPS base. For more information, please scan this QR code. Now, I briefly comment some of the results of this work. Several oncogenic pathways are deranged in MPS. When shown in Table 1, 9 of 31 pathways are enriched in all of the types of MPS analyzed, such as axon-guinas, focal adhesion, hypocynyl pathway, MAP kinase cynyl pathway, pathways in cancer, PI3K and AKT cynyl pathway, protoglycans in cancer, REP1 cynyl pathway, RAS cynyl pathway, and Wnt cynyl pathway. The most abundant pathway is the hypocynyl pathway, and the MPS type with the most enriched results are MPS type 7 or SLE disease. Also, several oncogenic pathways, such as cancer pathways, are found in the ontology terms. For example, breast cancer pathways, colorectal cancer pathways, gastric cancer, pancreatic cancer, prostate cancer, small cell lung cancer, hepatocellular carcinoma, and glioma pathways. Next, we see in Figure 2.1a oncogenic enrichment results of all datasets analyzed, and results of MPS type 7 enriched results when we see several pathways related to cancer enriched in these datasets. So, enzymes affected in MPS participate in the degradation of proteoglycans, such as heparan and dermatan sulfate. The proteoglycans interact with several signaling molecules in the cell. The primary function of the lysosome is degradation, but he also participates in other biological processes, like autophagy, cell adhesion and migration, cholesterol transport, and antigen presentation. This process also may affect in several types of cancer. Finally, the conclusions of this work: studying the tumor ontology signature in lysosomal disorders may help understand the mechanisms by which, for example, neurological physiology dysfunction occurs in patients with lysosomal storage disease and in neurological tumors. Besides, understanding this biological process may help us look for the new biomarkers for this rare metabolic disease. Thank you for your attention.